Hello, my name is Freya Cordell, and I'm a third-year undergraduate student working with Dr. Fred Andrus in the Department of Geological Sciences. Today, I'm talking about my work on a growth analysis of Saxidomus gigantea as a paleoclimate proxy for sea ice flux. This work is part of a much larger project called the Unalaska Sea Ice Project, which is dedicated to determining the feasibility of seasonal sea ice in the southern Bering Sea during the Holocene. In a world increasingly affected by anthropogenic climate change, we frequently discuss sea ice flux as one of the major results and indicators of a changing climate. Examining sea ice flux throughout geologic time is key to increasing our understanding of its relationship with climate and geosystems and building the efficacy of current climate modeling. The Holocene, a period that began about 12,000 years ago, brought on intense climatic shifts that allow us to study this change in great detail. My work specifically uses Saxidomus gigantea, a particular species of clam, as a proxy for understanding temperature and thus sea ice flux in the Bering Sea during the mid to late Holocene. This is Unalaska Island, a member of the Aleutian chain at the southern end of the Bering Sea. Sea ice has never historically reached this point, but in the early 2000s, researchers discovered the bones of several ice-dependent animals, including polar bears, beluga whales, ringed seals, and several others. These dated back to the Neoglacial period during the Holocene. Directly prior to the Neoglacial, the Aleutian Islands were even hotter than they are today, so the sudden development of sea ice would have been a truly shocking environmental change. According to Catherine West, an archaeologist who runs the Unalaska Sea Ice Project, this would be like Boston Harbor freezing over and polar bears appearing on our landscape. Therefore, the guiding question behind this project is whether seasonal sea ice did or did not reach Unalaska during the Neoglacial period. Crockford, the researcher who originally discovered those ice-loving animal bones, argues that the presence of juvenile members of the species provides incontrovertible evidence that sea ice must have been present during this time. The arrow on this map indicates her proposed extent of Bering Sea sea ice. However, the extremity of this climatic change necessitates different kinds of evidence to confirm that these bones came to Unalaska due to sea ice, and not for some other reason. And this is where Saxidomus gigantea comes in as another proxy. Saxidomus is a particularly effective paleoclimate proxy because of the nature of its growth. The species' growth is agreed by researchers to be primarily dependent upon temperature. It ceases at approximately 5 degrees centigrade. Growth increments also display tidal regularity, which allows us to directly infer time passage in the species' growth seasons. This image displays the key features of the clam. Winter cessation bands are thick, dark lines marked by A, B, and C, and the distance between two of these is a growth season, exemplified by D. E, F, and G are examples of those aforementioned tidal growth increments. Sample preparation is really important to making these growth patterns actually visual. So we start by cutting millimeter-thick sections along each shell along their axis of maximum growth, which you can see in the top image. Then these sections are polished, and we use something called mud-by solution to stain the samples and make the growth increments and cessation bands more visually distinct. And you can see a comparison of that in the second image. We finish this process by using a digital microscope to take high-resolution images of the shells. To begin data collection, we identify those winter growth cessation bands on the sample. Only the distance between the first and the second bands is measured, as this is the most stable year of growth for the clam. While the winter bands are usually easy to identify, factors like salinity changes or predation can cause stress reactions that appear visually similar on the samples. To address that, we microroll the samples to measure their delta oxygen 18 values. And as the graph on the bottom of the slide shows, higher values of delta oxygen 18 correspond with colder temperatures, allowing us to confirm that winter is the cause of a band. This graph displays the length between winter growth cessation bands of samples from our 3 archaeological sample groups and 1 modern site. The groups are ordered from oldest to modern, left to right, and you can see the geographic position of each of the sites on the map. Based on our data, average visual growth seasons were actually the highest in our oldest site, Margaret Bay, suggesting that growth conditions were the most optimal, suggesting higher temperatures during the earliest time period we measured, and not in the modern sample group. The other 2 archaeological sites did have shorter average growth seasons than the modern group, but not significantly. This is another look at the data, and as you can see, Margaret Bay, the oldest site, had the highest average growth season by more than a millimeter. Although the Amaknak Spit group did display a shorter average growth season than the modern group, this difference was only about 1/10 of a millimeter. Only Amaknak Bridge, the second oldest site, showed a significantly shorter average growth season of 7.884 millimeters versus the modern average of 8.77. Still, this difference of less than a millimeter may not be significant enough to support a climate change as drastic as the development of sea ice. To reiterate, the purpose of this work was to evaluate whether a growth season analysis of Saxidomus would support the feasibility of seasonal sea ice in Unalaska during the Neoglacial period. Theoretically, sea ice during this period would correspond with relatively shorter growing seasons in archaeological samples in comparison to their modern counterparts. However, the data indicate the longest growth season average in the oldest archaeological group and only a slightly shorter average growth season in the other archaeological groups in relation to the modern samples. While sclerochronology is one approach to understanding the feasibility of Holocene seasonal sea ice, the Unalaska Sea Ice Project originated due to zooarchaeological discoveries of ice-dependent animal bones. Recent analysis has definitively confirmed the presence of polar bones in Unalaska. Researchers argue that these bones are likely to be here as the result of nearby hunting or trading rather than farther sourcing, which suggests that these bones may support the presence of seasonal sea ice. However, the only site that bones were determined to unquestionably have belonged to polar bears was Margaret Bay, which is also the site we found with the highest average growth season. It's kind of unclear how to reconcile these findings. Many of the original bones provoking the Sea Ice Project actually belong to seal species. In particular, ringed seals are associated with year-round seasonal sea ice and bearded seals with sea ice in spring and summer. These species are the most prominent at the Amaknak Bridge site, which is the sample group that we analyzed with the lowest average growth season. So these coinciding pieces of evidence may support the presence of seasonal sea ice during this particular time period. However, it's worth noting that ringed seals specifically have been seen in recent years being forced to pup on nearby islands due to the lack of sea ice around them, complicating the certainty of any conclusions based on their presence. This could mean that alternatively, the presence of ringed seals at Amaknak Bridge was caused by a lack of preferred breeding conditions anywhere nearby. As this table displays, the Amaknak Bridge samples produce the lowest growth season mean. This mean is still less than a millimeter shorter than the mean of the modern group, but the correspondence of this data with the higher concentration of seal bones at Amaknak Bridge may be an avenue for further exploration of the possibility of seasonal sea ice. Further pursuit of delta-18 oxygen analysis and growth increment counts of these samples may provide more insight into this possibility, as well as confirming the cause of the high growth average of the Margaret Bay group, the oldest site. It allows the clam to grow more daily, widening the tidal growth increments and creating the illusion that a clam has had a temporally longer growth season. We can correct for this possibility by counting the growth increments themselves and verifying the number of tides that a clam has experienced. We're currently working on developing machine learning methods to automate the increment counting process to develop a more thorough dataset more quickly and to reinforce definitive conclusions about the role of Saxidomas in the Unalaska Sea Ice Project. Thank you all for listening. This is a brief acknowledgement to all of the groups and individuals who've contributed to this project, and now I will take any questions.
Why did researchers initially argue that seasonal sea ice was so likely to have been present during this time?
Their logic was that the presence of juvenile bones of the species meant that there had to be ideal breeding conditions, and most ice-loving species rely on sea ice specifically as a place to breed. So if juvenile bones are present, there must have been a place for them to actually have their young.
I'm curious whether there are other potential explanations for the presence of polar bones than neoglacial seasonal sea ice.
So the arguments of the researchers who discovered and have recently analyzed the bear bones on Unalaska were that the way that the bones were, the particular bones that they found meant that it was likely that the bears were killed or traded nearby as opposed to from long distances. But they only found 33 specimens in total, and only 6 of them were actually be able to be identified as specifically polar bears or brown bears. So it seems like from an outsider's perspective, they may need to do more work in definitively identifying the species of the bears, and also to rule out the possibility that Unalaskans, who were actually excellent kayak builders, didn't maybe go and hunt bears and bring them back via kayak versus some other overground method.
If growth increment counts create a more time-specific picture of the clam's growth, why wasn't this the initial approach to using the clams as a paleoclimate proxy?
So because there are so many growth increments in one season, manually counting them can take an incredible amount of time. and which is why we hope to optimize that process with machine learning and to just more efficiently be able to build that data set. But initially, we wanted to get a broader data set instead of that specific level of detail.
So I think it was on slide 10 that you had a column of, I think it was count, maybe it was another, where you had a table and you had, yeah. So that's the number of specimens, the count
Yeah.
column. So is that like all the specimens known or just the ones that were done in a particular study?
This is just the ones that were done in the study. We actually have more specimens to work with, but these are the ones that were dated, so kind of felt more comfortable starting with them.
And what is